Yep.
Okay. I think we can get started while people are still coming in. So welcome everybody to this month's BètaBreak edition. We're very happy to have you all here, including the speakers, of course. For those of you who don't know who we are, we're basically a student-run committee that organizes these monthly talks in which we invite a couple of top-notch researchers and we discuss a specific topic in a way that is accessible for all the students at UVA.
In today's discussion, we'll explore the role of computational methods in tackling biological issues across various fields, such as biomedicine and evolutionary biology. We'll also explore how these methods can help us transcend from some outdated practices, such as animal testing, but we'll also cover scalability and efficiency of these techniques. And finally, we'll look at some research that our speakers are currently conducting. So please welcome Dr. Shauna O Donovan and Dr. Wortel. And if you want to tell us something about yourself, your research.
Hi, welcome. I'm Meike Wortel. I'm an assistant professor here at the UVA. And I study microbial communities, so how microbes interact, for example, in a microbiome, so as in our gut live, and how they evolve. So, for example, how they evolve antibiotic resistance when you're treated with antibiotics.
Thank you. Does it, or maybe yeah. So my name is Shauna O Donovan. I'm an assistant professor in the Computational Biology group in TU Eindhoven. My research primarily focuses on developing modeling techniques and computational techniques to study human metabolism, and really specifically to understand what differences between people increase their risks for developing type 2 diabetes or cardiovascular disease, and if we can then use these models and computational tools to predict personalized treatments for these individuals.
Okay, thank you very much. So let's start diving into this topic. Of course, this edition is going to be about computational biology, but what is indeed computational biology? What do you actually do in your practice?
Yeah, well, in general, I think it's just using computational methods for biology. So this can be computer simulations, data analysis tool, machine learning. This is all covered in computational biology.
I would agree that it can be using computational methods to extract more information from your biological data. The data is getting bigger with omics levels, learning about your genome, the microbiome composition. We need computational methods to make sense of this, but equally using more knowledge to build models in a computer to simulate different situations and potentially use this for hypothesis testing. So testing different scenarios and seeing which one best replicates what you see in the lab or what you see in people.
Yeah, and what do you think that the fields of application are going to be in this specific field? Like, we're starting with small biological phenomena, but like, what are the possible fields in which it can develop?
Yeah, so I think computational biology, of course, has been used for a while, for as long as computers has been used. But it's really expanding with computers getting faster, more data being available, requiring more methods. In biomedicine, this is, of course, used a lot. Biotechnology, and also, my field, more evolutionary prediction, so for disease, pathogen evolution. And also for ecological applications. So, of course, with climate change, ocean acidification, we want to know and predict what's going to change there and how species can adapt.
I would say this is also where computational methods can be particularly useful. My own PhD was technically in systems biology, which is this idea of using computational tools to look at an entire system and not something in isolation that would be done in more traditional biological experiments. So the models we build really look at the whole human and how different organs interact and how dysfunctions in a particular organ affects what we can measure at the whole body level. And this is really where these computational models can be very powerful.
So also going off of that, does it provide a certain level of abstraction, or does it provide more of a holistic approach to the problem?
I primarily work with more bottom-up models. So these are models that we manually construct based on what is known in literature, so typically systems of equations. The level of abstraction is often the choice of the constructor. So our models are quite coarse-grained, describing largely what we can measure in the blood. So just changes in glucose or changes in insulin. In this way, we have a holistic model that looks at the entire system and how the liver, the pancreas, or the muscle give rise to what we see. But it's really at the choice of the builder. Equally, you have models that go down to individual transporters and individual reactions. So it's a choice at what your specific research question is, I think.
Yeah, I think all models are an abstraction of reality, of course. And if you can make models very, very detailed, trying to capture every aspect of reality, but then they also become as complicated as reality. So then you cannot use them anymore to see bigger patterns as well.
Yeah, I have a question that is not on the schedule. Let's see how you answer it. What is your approach with data? How important do you think that data is in constructing these kind of models? And how important is instead the theoretical framework that you have, of course, on this kind of phenomenon?
Yeah, I would say my focus has been more on the theoretical side of things, and less on the data. I think data is often a bottleneck in testing hypotheses. So with computational methods, we generate a lot of hypotheses, we generate new theory and create new insights. But yeah, I think let's also talk about the systems biology there, just the idea of cycle of systems biology. So you generate hypotheses, you use data to test it. So I think they're both important to get further.
Mm-hmm.
I think particularly as you move toward, I work in more biomedicine, and we would like our models to be used to inform treatments, and there having good quality validation is very important. So for us, having access to data, and it's not necessarily big data, it's just very good quality data from individuals to show that our models can replicate what is happening in the human, and therefore could go forward to be used for simulations of treatments. So in that respect, having good quality data, deeply phenotyped data is important for validation, but not necessarily for initial model construction.
And what does this kind of research provide that we cannot get with other methodologies, such as with lab research?
Such as?
In lab research, like more traditional techniques.
I think there are several things. I think, yeah, personally I really like when there's interplay between the lab research and the computational methods. Mm-hmm. And I think one thing where computational methods can help a lot is because of the scalability, actually. So you can, if we talk about microbial communities, if you want to do all combinations of a few microbes, this quickly explodes and you cannot do all these experiments anymore. Of course, there's also development in like high throughput lab methods, which is very nice, but yeah, it always remains limiting. And then the computational methods can help to select you the promising variants that you can then test in the lab. So they can very nicely interplay with each other. But I think the scalability is one thing that you can get. And the other thing is maybe you can have different systems that have a generic property, such as maybe like a feedback loop, for example. And yeah, of course, you can just look at all these different experiments, but if you can make a small model that shows the essence of a system and that it's applicable to many different systems, I think then you really learn something that you can use across fields.
If I may, we also use our models to get more understanding out of what has been measured. So our system of equations, we feed data into this. And this way we can reduce very high time series of data into a small set of parameters that we can more easily interpret. So a lot of the models that we have developed are used almost as a tool by experimentalists that we collaborate with to measure health in the data that they are generating. So it's not purely hypothesis generating. These models can also become tools that can be used by other researchers in their work.
So also, would this kind of research then be able to replace animal testing completely, or do we still need it to have like a how do you call that?
Like an in vivo test.
Yeah.
But I must say it's not directly my field, but of course computational methods, they will never exactly show life or exactly explain life or humans. But I think we have to take into account that animals also don't exactly represent humans. So their things go wrong. So I'm still hopeful, but I must admit it's maybe more Shauna's field.
There is a model in our field, so it's the DREAM model. It was made by a group in Italy and validated using very invasive data. But this one has FDA approval for simulating the glucose insulin system. So for manufacturers who are developing algorithms for an insulin pump, they can test their algorithm on this model in silico and skip animal trials. So in this way, you see that a really well-validated model, a good model, can drastically reduce the amount of animals, maybe not completely remove, but drastically reduce the amount of animals that are required for testing before clinical trials in humans.
Do you think, Meike, you mentioned that currently we don't maybe have enough like good models to completely substitute animal testing. So moving to Shauna with the same question, do you think at one point we're gonna like be able to create models with enough detail, but at the same time efficient enough to be more applicable to actually substitute animal testing, or is it like more of a theory that is never gonna be realized?
I think potentially more, as Meike pointed out, that you can carry out a lot more trials in silico. So rather than going into the lab and testing every possible combination or every possible dosage or everything that is a degree of freedom in your system, you could test many of these in silico and only go forward with those that show the most promising results. So in this way, you could really reduce what is happening. But I don't know if you would ever trust a model without animal testing to go straight to humans. I know in toxicology there's also a lot of work done looking more at in vitro models and developing more machine learning algorithms to translate what can be measured in vitro to what would be expected in vivo using human cells.
Mm-hmm.
Yeah, I think also models have to be even better than animals to be able to replace them. As in, we're used to the risks that come with animal testing and the differences. And of course, probably computational methods are going to have different things wrong. So if a computational method has something wrong that an animal test would have gotten right, I guess it will be judged harshly. So I think that will make it have some time, at least. But maybe never completely replace it.
Would you say that is the limitation of this kind of technique, or are there any more? Are there any more limitations to these techniques?
I would potentially say a big limitation is the availability of data. So that we can trust a model, we need to validate it sufficiently, and this is often a big limitation. I personally work and have very good collaborations with Wageningen and Maastricht universities, and we have access to data from human trials. So we know our models work well on a Dutch population of overweight and obese individuals, but how well this translates to people of different ethnicities, people of different ages, people with different health status. We need a lot more data and a lot more good quality data to be confident that our models can sufficiently describe all the situations that we would need.
So you still need some biological data, even from animal testing, in vitro, and human trials to compare your models to, right? How do you even compare them? Because even in vitro models, between each series, between each experiment, they differ. And even in vitro, you have 2D models, 3D models, organoids, all of that. So how do you even compare them when making a model computationally?
If I may, that's actually something we need. We need the replicates. I work primarily with human data, particularly something called a challenge test. So many people might be eating their lunch. We give people a very high-calorie meal, and then we measure their responses in the blood and how quickly you respond. So many of the young students, you should, within 2 hours, be back to what you were at fasting, and that's a healthy response. In clinical trials, this test is often done once because it's very expensive and it's very invasive. We haven't actually been able to test our model on multiple meals in the same person. So we have no idea what the variation just is day to day within somebody. So this is the type of data we actually need in human trials because we don't know what this inter-experiment variation is, just due to the measurement technique, or the day-to-day variation in a person is. So this is a big question for us in terms of how well our models describe the situation.
And for you, because you work with microbial organisms more, how would it compare?
Yeah. Well, we can. There's also a lot of variation, and I think often an issue is the condition. So often, well, you want to investigate how one parameter changes, like one thing, and so you need experiments with just one variable changed. Of course, the data is often possible to generate, but it costs time and effort. It's also, I guess, it's way easier than humans, so we have way more data. But then you also want to predict things in more detail. You need more data for that again.
Is it indeed nowadays cost effective? Is it cost effective to use this kind of methodology, so computational biology, creating this kind of models, instead of, for instance, in personal medicine, instead of animal testing? Is it cost effective so far, or is it still like in the process of becoming more efficient and cheaper, but it's still like only applicable to research or like, testing yet, but not in practice usable?
I think in the more biomedical application, the field that has demonstrated that this is possible is probably cancer treatment. So there was a big community effort in terms of generating data, a big community effort in terms of developing models and methods. I would say primarily more machine learning based models. And this has shown a great improvement in the prognosis for patients, where now we consider the genetic mutations in a particular tumor to inform which treatments should be given, where we consider other features of the tumor environment that can be captured to inform which treatment strategies should be used. And we have seen a great improvement in the prognosis. So I think this investment of time, resources, has shown that the application of computational approaches can facilitate personalized medicine. I'm not sure animal trials will aid us in personalized medicine. We really need more computational approaches.
Yeah, maybe another nice example where it can help is like for drug combination treatments. I think this is something where computational biology has shown. So often when you give one drug, a species or also cancer cells get resistant to this treatment. And then maybe it can help to give another drug. And sometimes when species or cells get resistant to a drug, they also get resistant to the other drug. But sometimes they get actually more susceptible to the other drug. And computational biology here has really helped to, or well, maybe this is still ongoing thing, suggesting drug therapies, how to combine drugs and how to alternate drugs to treat diseases. I think one nice example that came maybe a little too late is for HIV. I think in retrospect now we completely understand several different mutations and how we can treat them with different drugs. And well, it's, now of course there's different methods, so it's not that needed anymore, but it shows that there's definitely the possibility to do this. So depending on what kind of resistance mutations you find in a patient, you change the drug therapy.
And bridging from this to the next topic, what do you think that indeed is going to be able, like computational biology is going to be able to accomplish both in the personalized medicine and in bigger fields such as evolutionary predictions? What is it going to be able to accomplish in the next 5 years, considering data is more and more available every year, And of course, the models are going to be more and more capable, more efficient at handling data and at reprocessing. And yeah, I can also mention maybe AI is also going to play a role in this, I guess.
Yeah, of course, you hope that a lot will happen in 5 years, but it's in the end also not that long, so we'll have to see. So, in evolutionary research, I also want to add their data is, of course, a bit of a problem because you can measure a lot on bacteria, but in evolutionary research, you need the measurements over time again. This comes over evolutionary times, which doesn't have to be that long for bacteria. But still, yeah, it takes months or at least weeks to get these data. And then I think one nice example where there was a lot of data was coronavirus. With the pandemic, there was suddenly an enormous amount of data was shared. And here, well, there was a lot of discussions within the evolutionary biology community, like how will this evolve, how will this change? And it was still quite difficult. I mean, all the data was out there, but people didn't really know will become like, there's some theory that things become less pathogenic over time, but would that happen in this case? But I think it also did push the field a lot, also in the data sharing, like tens of thousands of genomes were shared very quickly over all the world. So what I think that's now really a changing field. So I do think there will be changes, but I'm a bit afraid to make really predictions about what will happen within 5 years. So there's several. So one thing, evolutionary predictions are used for influenza. They're for the vaccines. So which vaccines are used in the next year? And I think there it's nice because both data is used from different countries about what strains are persistent, but also evolutionary knowledge is used there, like which strains can evolve quickly, and where do we expect the next virus outbreak to come from. And I think this will increase, but yeah, how that will be a tangible difference, that's...
So do you think that maybe in 10 years or so, instead of 5, we could be able to predict with like a decent accuracy an outbreak or COVID, of new COVID or, you know, some kind of virus like that? Or is it more still of a utopia for maybe 50
I think
years?
it will be hard to predict a new outbreak where it will come from, but maybe as soon as an outbreak starts, it will be better to predict like how big it will be or what kind of vaccines would help or would...
How to tackle it, of course.
Maybe another thing is digital medicine, which has been a topic recently. Maybe would it help in the epidemiological predictions?
Sorry, I didn't hear.
I said digital medicine has also been a big topic, so would that maybe help with the data sets and the predictions for epidemiological outbreaks?
So maybe...
It's not directly related, but indeed I'm more familiar with what's happening here in the Netherlands. There's a lot of infrastructure and cooperation between the hospitals and a lot more openness with sharing anonymized data sets with researchers. I know in Eindhoven we work very closely with the medical centers in the city where they have used patient databases, developing an algorithm to predict risk for COVID, to try to optimize patients coming in, who should be tested for COVID, who should not. And they could show that when they applied this in subsequent years, it was good for saving costs, that they could better predict who needed the COVID test and who didn't. Equally, they use this data for developing algorithms to predict cancer risk using measurements collected in blood rather than the need for more invasive biopsies. So there is greater standardization of these databases, at least across hospitals in the Netherlands. And there's resources supported by SURF that allow for researchers to access this data in a safe way that complies with GDPR, which then allows them to better train models for the prediction of diseases from various medical records and decision support systems. I know Philips is also involved in several projects in Eindhoven to support clinicians in making decisions on when patients, for example, in ICU may need interventions and when they may not. But these are really decision support, so the decision always remains with the clinician, but we can provide them with additional metrics and scores to support them in making these decisions.
I guess also, of course, you mentioned the GDPR. I guess, of course, privacy, especially in the case of personal medicine, plays an important role in how we deal with data, how we collect data. Do you think there's a way to get a lot of data, enough data, of course, to make good predictions on how to cure patients and on disease in general, complying with, of course, the privacy of the patients?
It is possible, but it's a big headache. So indeed, you would need every institution to agree. The big challenge, which is somewhat being overcome, is the standardization of data. So typically different hospitals or different research centers would store their data in their own way, or they have their own policy on which tests should be performed. So you have a lot of missing data or incompatible data. This is being standardized more and more, which allows you to combine data from different centers, but anonymized, and the data is typically still housed in the medical center, in the hospital. There's a lot more also federated learning, which are machine learning algorithms that are then specifically designed to, the algorithm itself goes to the center and trains on the data. The data doesn't become centralized. So in this way, the data doesn't directly need to be shared, but the models can still be trained and learn on that information. And I think that's potentially where the field is going, more so than sharing the data because of GDPR.
And so far, another issue, of course, in the collection of the data is where it comes from, I guess. Do you see in your fields of research that like most data comes from European countries, or is it already trying to develop in a way such that like developing countries as well, and I guess the continents of Africa and Asia maybe, that are usually less represented, we can gain more data from these places, or?
It is a big challenge. So as I mentioned, often it's a choice made for study design. So many of the clinical trials I get data from, they specifically choose to only include people of Dutch descent to allow them to work with a smaller sample size that they can afford to measure in. So this is often a choice currently in study designs, but it does mean that the models trained on these data sets, we don't know if they generalize to other populations. There is a lot of pressure, and rightly so, to try to diversify these data sets, to try to test them on other study populations. Particularly data sets coming out of the US and the UK tend to be a lot more diverse. And this is a great benefit to testing and validating these models. I think definitely there needs to be a lot more investment in diversifying the data sets, not just for source or ethnicity, but particularly in terms of men and women. So there are equally many trials that only include men for simplicity, but then that means that we don't necessarily know if our models can describe what's happening in women at different stages of the menstrual cycle, postmenopausal. We know all of these can have an effect on glucose, so my own field, but we don't account for them in our models because we don't have data on this. So there is a real need, if we want these models to really go into clinics eventually and to inform clinical decisions, that we have tested them on sufficiently diverse populations. But I think we're only starting to get there.
Mm-hmm. Maybe to add a bit to that, so I study microbial communities like the gut microbiome, and also there the first studies were also less diverse, although there's now some improvement. And also in Amsterdam there's also called a multi-ethnic cohort where they especially looked for simpler sample sizes with different ethnic backgrounds. So indeed, they're starting to get some benefit. We are hoping to, with the microbiome models, to predict also interventions. And it can also be possible that at some point can be in a personalized way. Like you could get someone's microbiome composition, someone's genetic factors maybe, and then predict what kind of interventions could help with diseases. But yeah, we are not there yet, so I cannot say how that's going. Yeah. But maybe that's for the next 5 years.
For the next BètaBreak edition in 5 years. Anyways, if there's any questions already, we can take a couple questions before moving on to the specific research fields of the researchers. Otherwise, yeah.
There's a question.
Oh.
Yeah, oh, damn, sorry. I was just wondering, how would you go about selecting a data set when you're choosing between one that is more representational versus one that is more abundant? And like, which considerations come into play when making such decisions? That's my question.
Yeah, so I think we often don't have so much choice.
Right.
We are happy with all the data we can get.
And if there was choice, I think it depends on your research question. So often if we build these more bottom-up models with equations, we would prefer one person who was super deeply phenotyped than 1,000 people. That if we have the measurements of the internal fluxes or reactions that we need, that is better than having 1,000 people. If you want to go to more machine learning algorithms, they're having more people and capturing this inter-individual variation in your model is more important. So if you do have choice, often it's based on your research question. What data allows you to test your model the best?
Thank you.
Any more questions?
What are some of the mathematical fields your research entails?
So I work a lot with differential equation models, and I think it's similar. So yeah, but also a lot of optimization we use because, well, evolution often when we're trying to understand it computationally, we kind of look for in a certain way an optimum, because evolution leads to better adaptation to the environment at least. So you can look for states of the model where it's optimally adapted to the environment. Therefore we often analyze sets of differential equations, but also sometimes stochastic effects. And for that we also use simulations.
Equally, we primarily use systems of ordinary differential equations to describe the biological system. We focus a lot on methods for data assimilation, so how to personalize the models by fitting them to measured data and testing the quality of that and uncertainty. In my group, we also look a lot at more machine learning algorithms, some small deep learning networks or traditional statistical approaches. And increasingly, we're looking at how to actually integrate these so that we can have a machine learning approach that has this flexibility to learn from data, but that it's constrained by knowledge using the ODEs. So the ODEs will encode really the biological laws. We know this happens, and this leaves the machine learning algorithms free to learn, hopefully, the missing parts. So that's where we're currently moving.
Thank you.
So back to your research. Shauna, so you research more personalized medicine, and you mentioned that you have developed some of the new methods with hybrid modeling, utilizing AI. Would you want to tell us something about that?
So these are the methods that I just kind of alluded to. I think in most fields there's this big AI boom that it will solve all of our issues. But the big challenge is these models require huge volumes of data. I think there's some very famous groups in the US and Israel who are really trying to apply these models to learn this glucose response, but they have data sets of tens of thousands of people, and they're still not getting very far because there's so many factors that influence these glucose responses. So we're working on these hybrid models, which have been very successful in different more physics and engineering-based disciplines, where we're really taking these machine learning models and embedding them directly into systems of differential equations. So as I mentioned, the differential equation takes care of what we already know in the system. So that insulin will affect glucose, that when you ingest a meal, the glucose will go up, with the idea that the machine learning algorithms can learn the parts we don't currently know. And we've been testing these on more simple toy examples where we kick something out of our model and we see if we can learn it back. And so far, they've been able to work quite well. So now we're looking at taking these and applying them to learn how the microbiome composition may influence these responses by having multiple meal responses to different meals and the microbiome composition, and learning what components of this composition influence this glucose response, knowing that how much protein and how much fat is in the meal also plays a role.
Yeah, and I have a question about more or less the timeline of the research. So you have this hypothesis, we want to measure the insulin in the patients. We do this, then we create a model that represents and replicates the whole process. What's the timeline in between, like, hypothesis, the actual model creation, of consultative recollection in the meantime, and then we found something, we're going to be able to use it for patients from this moment onwards? What do you think more or less is, yeah?
I will say we haven't completed the cycle in terms of getting it to patients yet, because we haven't had the data to validate it to that extent. In terms of physically building a model, the last one I personally made took 18 months, nearly 2 years. So this is partially why we're excited about these hybrid approaches, because it's not feasible to manually build the model based on the known biology, or at least it's not something I think could be done in my lifetime if I sat down to do it by myself. So it does take quite some time of testing different hypotheses, going to literature. It's not a fast process. And we also don't collect the data. So the data is already collected. We have a hypothesis. We build our model, typically taking over a year, if not more. And to get it to the stage where it could inform clinicians, I think could be 5 to 10 years. A bonus is our models are being used by researchers. So they are currently being used in clinical trials, but not at the stage of really being rolled out in a clinic.
And do you think that it's going to get faster throughout time when we have like more models, we already know the approach to use and stuff, and also maybe with the sharing of information and knowledge between universities or research centers?
I think for us, all of those steps, so these hybrid models can hopefully provide us with a way to accelerate model development. And if more data is available, it will then allow us to validate the models and move through these steps more quickly. It can be very difficult to access data. So I'm very fortunate that we have strong collaborations with some Dutch universities who are willing to share data with us. But for privacy reasons, this is still not a very fast process. There's a lot of agreements that have to be put in place. The study participants' privacy must be protected. So it's not a very fast process either.
Mm-hmm. Yeah, and moving on to Meike, your focus of research, at least in previous times, was evolutionary predictions. We already started to talk about the evolution of viruses, and of course that's way faster than the actual evolution of mammals or humans even. So let's start from the evolution of viruses. What can computational biology do for studying these kind of evolutions?
Yeah, so there's one thing I didn't really touch upon about predicting evolution, because for now we have assumed that it's something we should understand and then we can predict. But evolution is a combination of the generation of variation and then the selection on it. And this generation of variation is often from mutations. And mutations, there's some stochasticity in what mutations happen. So I think there's maybe two limitations to predicting evolution. One is the availability of data and knowledge of the system. And one is the fact how predictable it actually is. How big is this stochastic component? And probably it's not can we ever predict or can we never predict, but to what extent can we predict things? So what's this inherent stochasticity? So that's one thing we also study. Like, for example, what you could do is, you can have the same thing evolve it twice, and do you twice get the same outcome? Like with a deterministic lab experiment, you of course hope to get the same outcome, and maybe there's some noise on your data, but in this case it could be that, that you just keep getting different outcomes, and then, it will be very hard to predict. Still, I think we think there's definitely limitations to the predictability, but there's also things we can predict. I don't work a lot on viruses myself, more on bacteria, but so viruses are faster. They, of course, I gave some examples that people try to use predictions in there, but maybe moving on, maybe I can say a bit more about bacteria. Mm-hmm. And so there we try to, they still evolve pretty fast, so we can do them in a lab experiment. Especially with antibiotic resistance evolution, I talked about this variation and selection. So with antibiotics, you have a very strong selection. So within weeks, you see dramatic responses of bacteria, sometimes even faster. And we can try to predict how these happen. And one very interesting thing I study is that bacteria evolve, but by evolving they change, but they also change other things. For example, some bacteria change to break down the antibiotics, and then there's no antibiotics anymore, and then other bacteria in principle could grow again. And these things might also affect evolution, and does that help us to predict evolution, or does it make it more difficult? So, that's what we are hoping to find out.
And do you also think, maybe not now, maybe in 10 years, maybe in 20 years, I don't know the timeline, but do you think we're going to be able to predict something about humans or like mammals? We're going to have enough computational capabilities.
Of course, indeed. Well, because we were talking about this, so there's a famous experiment with bacteria that started in 1988, I think, and they're up to 80,000 generations now. Of course, with humans or mammals, that will take a long time. I don't think humans are evolving that fast. But in other species, you definitely see effects also in larger species. And one of these things I already mentioned is changing climate. So people are studying a bit like how can species adapt? Do they move or can they adapt more easily? And also, one nice example, although it's not so clear how strong the effect of evolution was, is in fish. So sometimes people make rules that you can fish only up to from a certain size, so you cannot fish the small fish. And then people studied if you actually see a change in fish, that they start to mature at a smaller size, because the larger fish gets fished away. This gives the selection pressure, and you see changes in fish populations. It's not completely clear in how far this is an evolutionary adaptation, but so definitely in larger animals you can also it's also studied. For humans, I don't know how big the effect of evolution is, recently at least. So it's more studying what happened in the past. Mm-hmm.
Also talking about the future, is it possible, since you research microbiome, and Shauna also mentioned that you research how microbiome can affect insulin resistance. Is it possible to combine these models? How are they made? Are they made to be shared?
I think we do try to share the models that we make of the microbiome and the insulin, but maybe...
Yeah, definitely. We want people to use our models. So there's field standards. There's a particular language, SBML, where you can... I personally don't know how to use it, but someone helps me to put my model into this language, where you can open it in multiple software in a standardized way to support sharing it with other researchers. We always make our code available. We encourage people to use it. It probably takes a bit of puzzling the jigsaw piece together to directly combine models, but if they're systems of differential equations, in principle, it should be achievable.
Yeah, I agree. I don't think models are things that people often want to keep to themselves. It's more something we really
And yeah,
share.
for you as well, Mike, in your field specifically, what do you think that is like more or less the timeline where you're at in the research, and when is it gonna be, when is it gonna be usable in practice for instance, for antibiotics predictions and stuff?
Yeah. Yeah, so one thing that would be perhaps feasible, so I try to look at how antibiotics change the evolutionary, maybe at some point you get less resistance evolution, or you can reverse it. Still, I don't work on the applications myself, so there's definitely, I would say, a 5 years of someone using these insights for application. The research on microbiome, hopefully quicker, so we're in a big consortium also where we have a cohort and we try to make models, so we model all the microbes together, and then we look at certain effects, so both in the lab and for us on the computational side, so what if you eat more often sugar, how does that change? Could you add some compounds so that the community changes to your benefit? I think if we find things, typically it's a consortium where also the implementation of these interventions is part of not the implementation itself, but the suggestion and the study of how to implement these things. Yeah, hopefully we can at least say something within 5-10 years that could be helpful.
And yeah, one last question before we leave space for the audience. Do you think this is going to be the way that your research is going to go, or like computational biology all the way, that's how we're going to go models from now on? Or do you think some alternatives are going to pop up? Maybe, I don't know what. Yeah.
I think for me, my research will always be computational. I'm a mathematician by training, and from my bachelor's degree, I've always been fascinated how systems of equations can describe a biological system. And I find it very rewarding or satisfying to see people use my models with their data, or to see what insights my models can give. So for me, for that more personal reward, I would stay with computational biology. I think there's a huge amount of potential there in the field. Maybe I'm biased, but I think the way data is growing, it's getting bigger in biology. Experimental design is getting more complex. We need computational methods to be able to handle this. It isn't possible using some of our traditional t-tests anymore. You need newer methods, newer models to be able to get insights out of this data. So I personally believe I will stay in computational biology, but I think generally as a field, we need to invest in computational biology in order to be able to address these bigger biological questions.
Yeah, I agree with that. Personally, I also think I stay in computational biology, but I'm not as strongly on the computational side. So I also have a background in biology, and I really like also this combination of experimental research and models. How can that interplay? So I think, it's not computational all the way. Maybe not for me, not for as we talked about. You have this cycle of hypothesis generation, some tests, and then the data analysis to come to new hypotheses. So I think it's always going to stay an interaction, but this interaction also needs computational biology. And indeed, with the amounts of data and the more complicated data and the bigger systems we're trying to study, it will become a larger portion of biology, but it will never replace all the rest.
Follow-up question. So far, I think most of the research in general, the approach is being, we need more and more data. Do you think this is the way that we're still going to go, or are we going to concentrate on the quality at some point, more than on just adding more and more data to our models?
I don't know if I'm the person to necessarily answer, because I am a mathematician. So we collaborate with people who generate the data. I think there needs to be, definitely coming from the field of more personalized medicine, there needs to be a more coordinated effort either across the Netherlands or across the EU to standardize the type of data, or more so to perform larger trials. Multi-center trials ideally, where we can get this diversity in, but where the type of data that is being collected can be standardized in order to feed the models. Currently, many studies fit into a PhD cycle. So it's this 4-year cycle where they have time and money to measure maybe 100 people well, looking at a specific hypothesis. And then in the next 4 years, they have a different hypothesis. And in the next 4 years, a different hypothesis, which means we never get sample sizes bigger than 100 individuals. I think if you look at the US, if you look towards these groups in Israel that are generating tens of thousands of people worth of data, that is what we need if we are to really advance personalized medicine, that it's good quality, standardized data that's coordinated. It's not possible for one group or one university to generate this. It needs this coordinated effort. And if that's somehow enforced by a granting agency that says, okay, you have to do these type of measurements. But I think that's what's needed to really, if we talk about advancing the models towards clinic, it's that type of data that's needed. Good quality, standardized data.
Yeah, I think there's also a difference in what's going to happen and what should happen, maybe. I think there's often a big reward to generating a lot of data. I mean, if you generate a lot of data, you have something out of your study, that's not, it's not always useful to just generate more data. But I do feel that there's starting to get a little bit of a change in the field. More people are saying, like, we need to reuse data, for example. So I think also grants just reusing existing data become more having a bit more chance to get funded. And there's also people, we really need theory behind all these data. If we just generate data, we get lost in the data if we don't have a clear direction of where we're going. So I feel like it's a small start of a little bit of a shift of not just generating data, but also indeed looking at the quality and also looking at where it's going.
Yeah. Thank you very much. So this was more or less what we wanted to discuss with you. If the audience has any questions for the speakers in their specific field or anything.
Hello. Yeah. Thank you for your insight, Dr. Shauna O Donovan. I wanted to continue about the database and the data collection. I ran into problems myself with the collection of data and not being similar. And what I remember is within the neuroscience, they kind of set up a larger database. They were starting to work together. My question to you now is, how do you envision, or the brain is so big, can we find something within that? And how do you think we can maybe calculate something within that? Yeah.
Well, I'm not as familiar with the brain, but if we think of these databases, if they're standardized, a big challenge coming from our field is GDPR. So our data is actually never made publicly available. You have to go to the specific group, ask, make arrangements, usually working together to analyze the data. But you do see in fields where there have been efforts to create these larger databases that it allows computational groups that are typically smaller to access data that they otherwise wouldn't, and potentially find new insights in trying out a model or trying out a method. And I think in those fields where you have these big publicly available data, also bioinformaticians with publicly available transcriptomics or genomics data, it has allowed a lot of methods and tools to be developed that otherwise couldn't. So I think these standardized public databases with sufficient metadata can really allow the computational biology fields to grow. But it does depend on the field. So for us, the data is very rarely actually made publicly available.
Thank you so much.
Hello. I'm wondering, like, for the data that you were talking about, like, for collection and, like, making, like, the data that you collect in, like, maybe clinics and stuff, like, would that be, like, data that's, like, pretty expensive to collect that would involve, like, complicated scans, or is it, like, the regular data that, like, all of us already take, like, in clinics? Thank you.
It's a mixture. So within our group, there are people who work on projects that really focus on building models that use standard clinical data from the hospital. So standard blood test, the information that is coming in maybe from sensors that are put on the body in the ICU. So there are projects where they're really focused on supporting what is currently going on in clinic. For myself, the models we're developing are a bit more exploratory, and we use data from clinical trials. This data is typically very expensive, and the data we would like is something called stable isotope tracers, not to be too detailed, which involves ingesting glucose or fat that has been labeled in a way that it can be safely consumed by people, but it is incredibly expensive. So that's what we would dream for our models. But the cost associated with doing these experiments means that the data is very rare and very small. But that's enough for us. But then those models have been developed for very different data. So again, it depends on the project and the question. For model development, you often want this very expensive data, but typically once it's validated, you can then aim to translate it to data that can be collected maybe with a wearable sensor. So currently there's a lot of interest in these. You might have seen people with these wearable glucose devices on their arms. So we're now trying to get our models that we validated on very expensive data to work with data that can be collected at home in real life conditions.
Thank you so much.
Yeah, I also have a small question for you. So in most fields in technology, we've seen China especially, but also the US being like the lead in the research and in like, of course, the funding that they have. Do you think that it is also the case in computational biology, or do we still kind of have the European primate in this kind of research?
I think for the moment it's not really focused in one particular place. Of course, in the US especially, there are lots of big research institute universities that work a lot. But there's also certain big places in Europe, for example, that are strong in computational biology. I don't think so much there's that much from China yet. Maybe it's also not the interest.
Yeah, it's the same. I think from the more computational perspective, for us, some of the bigger groups are still based in Europe. But it's more indeed often there's one PI who started working in glucose insulin modeling in the 70s. So their group is still one of the most prestigious or that have the greatest output at the moment. But I don't think it's as regionally distinguished that way. In terms of data generation, you do see the very large cohorts are coming from the US, coming from Israel. Really, there's been a huge investment there. But increasingly, there's companies, private entities coming on board. So for this personalized nutrition, there's a company in the UK called Zoe where they have data now for 100,000 people. And they are willing to test models and different prediction algorithms on their data. So I don't think it's as regionally classified that way. Yeah, the models are coming out of everywhere, to be honest.
With that said, thank you for coming. We were very glad to have you here. And we'd like to introduce the next BètaBreak edition, which is on blockchain, which will be in February. And thank you for coming. Hope you enjoyed this panel.